Hello students, I am Shahin Mahmood, currently studying MBBS from College of Medicine and GRM Hospital Kalyani, and today I will explain the botany part or botany section of the full test 10. So let's start with the first question, section A. It is saying that food material which is very similar to amylopectin and glycogen in structure is stored in food of rhodophyceae, phaeophyceae, chlorophyceae, or cyanophyceae. As we know that in rhodophyceae, the food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. So basically, what is the thing or what is the food stored as in rhodophyceae, that is Floridian starch. Floridian starch. So first option will be your correct answer for this question. Moving towards the next question. Gemma are green unicellular asexual buds which develop in small receptacles called gemma cups located on the thalli. Reason is given. In liver oats, spores germinate to form free-living sporophyte, and we have to choose between these four options. So first of all, if we check the first statement, it is saying that gemma are green. It's correct. Unicellular. Gemma are not unicellular. Gemma are multicellular. Unicellular is incorrect. Then asexual buds which developed in a small receptacle. Rest statement is correct except this part, unicellular. This whole statement is wrong according to this. Next, it is saying that in liver oats, spores germinate to form free-living sporophyte. So basically, in liver oats, spores germinate to form free-living as not a sporophyte but gametophyte. So this is also wrong. This word is wrong. It will be gametophyte. So both options are wrong. Both assertion and reasons are wrong. So fourth option will be your correct answer if both assertion and reason are false. Moving towards the next question, that is saying that ground tissue includes all tissues external to endodermis, all tissue except epidermis and vascular bundle, epidermis and cortex, all tissues internal to endodermis. Before going to the answer, let us understand what is ground tissue. So basically, ground tissue can be defined as all the tissues except epidermis and vascular bundle. So ground tissue constitutes the interior of organs except vascular system. It consists of simple tissue such as parenchyma, then collenchyma or sclerenchyma. So what is the work of this ground tissue? Ground tissue basically support the system, support the plant structure. It also helps in store molecules. Then it also helps in photosynthesis. So these are basically the functions of ground tissue. So yeah, if we consider this question, according to this question, all tissues except epidermis and vascular bundle will be your correct answer for this question. Moving towards the next question, 104. It is saying, which of the following is not a stem modification? Rhizome, corn, feature, or tuber? So rhizome is a stem modification, corn is a stem modification, and tuber is also a stem modification. We know that tuber you can see in cotton. But this feature, what feature basically? Feature is basically leaves of certain insectivorous plant, such as feature plant, Gnus flytra. These are basically modified leaves. Modified leaves. These are not the modifications of stem. So third option will be your correct answer for this question. Moving towards the next question, 105. It is saying that spores in Funaria produces from foot of sporophyte, seta of sporophyte, capsule of sporophyte, or all of these. So basically spores in Funaria. Funaria means what does Funaria is? So Funaria is basically moss. Funaria is moss. and it produces from the capsule of sporophyte. The third option will be the correct answer for this question. And these spores are basically the first cell of gametophytic generation. Moving towards the next question, 106. It is saying a special type of structure is found in brinjal where stamens are attached to the petals. Name this condition. So this special type of condition where stamens are attached to the petals, these are basically called epipetalous. The second option will be our correct answer for this question. If we consider or if we understand, if we want to know the meanings of these other options, epiphyllous, gamocephalous, gamopetalous. So gamocephalous means when sepals are fused. Gamopetalous means when petals are fused. And epiphyllous means when this stamens is attached to perianth. That is epiphyllous condition. So that's why considering all this, second option will be our correct answer for this question. Now moving towards the next question. 107. Read the assertion and reason carefully to mark the correct option in the question. It is saying pili and fimbriae are known to help the bacteria to become motile in streams. Next, it is saying that reason, unlike flagella, pili and fimbriae are surface structure of bacteria, and we need to choose between all these four options of assertion and reason. Basically, if we want to consider this, pili and fimbriae are known to help the bacteria to become motile in stream is absolutely incorrect. So what does pili and fimbriae does? So basically pili and fimbriae are known to help the bacteria to attach with rocks or any other substances. It doesn't help in motility. And reason is given, unlike flagella, pili and fimbriae are surface structure of the bacteria. So pili and fimbriae are the surface structure of the bacteria. This is correct, but unlike flagella, flagella is also a surface structure of the bacteria. That's why this reason is also wrong. So both assertion and reason are wrong. That's why fourth option will be our correct answer for this question. Moving towards the next question, that is 108. Which of the following scientists explained the cells derived from new cells are formed from the pre-existing cells? So basically options are given: Schwann, then Schleiden, Robert Brown, or Rudolf Virchow. So basically Rudolf Virchow explained that the cells derived from new cells are formed from pre-existing cells, omnicellular, acellular. So this is given by Rudolf Virchow. Fourth option will be our correct answer for this question. Moving towards the next question, 109. The characteristic feature of diplotin stage of meiosis first is recognized by dissolution of synaptonymal complex, homologous chromosome of bivalent structs separate from each other, egg-shaped structure called chiasma are formed, and all of the above. So let's consider one by one. First is saying a diplotin stage of meiosis 1. Okay, that means this is basically a stage in prophase 1. So first option is given, dissolution of synaptonymal complex. So yes, in diplotin stage we can see dissolution of synaptonymal complex. So this is the correct statement. Next, homologous chromosomes of bivalent structs separating from each other. This is also correct. And then it is saying that X-shaped structure called chiasmata are formed. This is also correct. What is chiasmata? We know that. We want to make a diagram. This X-shaped structure is forming X-shaped structure. This is called chiasmata. So all these options are correct. So fourth option will be our correct answer for this question about diplotin stage of meiosis first. Moving towards the next question, 110. In meiosis first, prophase 1 subdivided on the basis of meiosis first, prophase 1 is subdivided on the basis of chromosomal behavior, chromosomal size, chromosomal number, or chromosomal structure. So fourth option will be our correct answer for this question, because we know that how this chromosomal structure change between these phases of prophase first. And if we want to see that, we can see that the nuclear membrane and nucleus reappear, cytokinesis follows, and this is called the diet of the cell in telophase 1. And apart from that, we can also see characteristic feature of chromosome in different parts or different stages of this prophase first. So moving towards the next question. A bivalent consists of 2 chromatids and 1 centromere, 2 chromatids and 2 centromere, 4 chromatids and 2 centromere, 4 chromatids and 4 centromere. So basically, what does a bivalence? First of all, we will draw the structure and we will try to answer. So, this structure will consist of bivalent. You can see that 1 chromatid, 2 chromatid, 3 chromatid, 4 chromatid, and 1 centromere and 2 centromere. So 4 chromatid and 2 centromere will be our correct option for this question. If we want to know what does bivalent is, so a complex formed by a pair of synapsed homologous chromosome is called a bivalent or a tetrad. The structure here I draw is just before the bivalent. These are basically the homologous chromosomes. So if we want to make the actual structure of bivalent, which is this, the chiasmata-form structure, we can make like this. This is basically the proper representation of bivalent. And we can count from here as well. Third option will be our correct answer for this question. Moving towards the next question, 130. It is saying that in C4 plants, photorespiration takes place in mesophyll cells. Next, photorespiration takes place in bundle sheath cells, and then photorespiration do not take place due to maintenance of high CO2 concentration at enzyme active site. Next, it is saying that both 1 and 2. So we have to understand this question that in C4 plants. So C4 plants have basically a special mechanism that increases the concentration of CO2 at the enzyme site, and CO2 fixation is done by Rubisco enzyme. And we know that these enzymes have 2 types of activity. One is carboxylase, one is oxygenase. Carboxylase and oxygenase. In C4 plant, it ought to be carboxylase activity, and that's why photorespiration does not take place due to maintenance of high CO2 at the active enzyme site or enzyme active site. The third option will be our correct answer for this question. They have a special mechanism. In C4 plants have a special mechanism which increases the concentration of carbon dioxide or CO2. You may ask that why this CO2 increases? What's the reason behind this? The reason behind this is that this basically happens because mesophyll C4 acid broken down. So mesophyll C4 acid broken down in the bundle sheath cell to release this CO2. That's why the concentration of CO2 is higher in C4 plants. So third option will be a correct answer for this question. Moving towards the next question, 140. It is saying that when a pollen of a flower is transferred to the stigma of another flower of the same plant, the pollination is referred to as autogamy, hypnogamy, xenogamy, or allogamy. So apart from all these options, we will try to explain each and every one. So basically, what does it mean that hypnogamy involves the transfer of pollen from one flower of a plant to the stigma of another flower of the same plant. So you have to remember this is for same plant. Same plant. For example, maize we can see. So as the pollen has to move from one flower to the another flower, it requires a pollinating agent. Yes, it is genetically similar to autogamy. Both the flowers of the plant is same because they have the same plant. The genetic composition is same, and they have the same genotype, but this process is known as geitonogamy. Second option will be your correct answer for this question. Now moving towards the next question, which is 150. It is saying that in which of the following plants only chiasmogamous flowers are present? Pea, rose, pansy or peanuts? So basically we have to understand that what is this chiasmogamous flower and what kind of flowers are present in a plant. So we have to understand this before going to mark this option. So basically this chiasmogamous flowers means that flowers that open at maturity. Flowers that open at maturity. At maturity to facilitate the exposure of pollen and stigma for cross-pollination and referred to as chiasmogamous flowers. And they are basically large, colorful, and along with that we can also say that they also have nectar or scent to attract pollinators because it opens to have cross-pollination. Now there are another kind of flowers as well that is called clistogamous flowers. The clistogamous flowers basically remain close to avoid cross-pollination. For this question, a rose is the chiasmogamous flowers among all these options. The second option will be your correct answer for this question. Now moving towards the next question, 100. It is saying that if a cross is made between F1 progeny, pink colored flowered plants and white flower bearing snapdragon plants, what will be the percentage of red flowered plants in their progeny? And options given 25%, 100%, 50% and 0%. So after reading the question, you can notice that one specific plant is given, that is snapdragon plant. And we know that this case of snapdragon plant, this shows incomplete dominance. Incomplete dominance. So what does it mean? We'll try to explain. So basically snapdragon shows incomplete dominance. It means that one allele for the flower color is not completely dominant over the other one, and thus heterogeneous genotype shows the combination of dominant and recessive phenotype. The genotype of pink flower is RR, one is small, one is capital, so it shows that it is heterozygous. It is heterozygous, and that's why it's have pink color, not red, not white, intermediate pink flowers. Now the genotype of white flowers is this. So the cross will made, it will for this one. Therefore, pink and white flower are in equal proportions. 50% pink will obtain and 50% white will obtain. Percentage of red flowers will be 0. So the percentage of red color flowers will be 0. Because there will be no red color flower as pink and white color flower will be there because of incomplete dominance. So fourth option 0% will be your correct answer for this question. Moving towards the next question, 107. It is saying that select the incorrect statement. All 3 RNAs are needed to synthesize a protein in a cell. Next, it is saying that rRNA play catalytic and structural role during translation. Then tRNA reads the genetic code along during translation, and then lastly RNA polymerase second synthesis hnRNA in prokaryotes. So let's explain one by one. All the 3 RNA, that is rRNA, mRNA, tRNA, are needed to synthesize a protein in a cell. We know that. This is the correct statement. Next is rRNA, that is the ribosomal RNA, play catalytic and structural role during translation. This is also correct statement. Now the next is tRNA tRNA reads the genetic code during translation. We know that these have the codon, so anticodon sequences, so it reads the genetic code. in the tRNA structure. You will see somewhat like this. You can appreciate this. It is somewhat like the rough structure of tRNA. You know that this has the codon, anticodon sequences. And then lastly, it is saying that RNA polymerase second synthesize hRNA in prokaryotes. This statement is wrong because the RNA polymerase second transcribes precursor of the mRNA, the heterogeneous nuclear RNA in eukaryotes, not in prokaryotes. It should be in eukaryotes. This statement is wrong. The fourth option will be a correct answer for this question. Moving towards the next question, that is when a single gene influences more than one trait is called pseudodominance, pleiotropy, epistasis, or none of these. So basically, among these four options, we have to choose. So basically, what happens that the ability of a gene to have multiple phenotypic effect because it influences a number of characters simultaneously is known as pleiotropy. So the ability of a single gene, and this is influencing multiple characters. That's why this is pleiotropy. And the best example of pleiotropy we know, that is phenylketonuria. So second option will be our correct answer for this question. Moving towards the next question, 190. Who proved that DNA is a basic genetic material by giving unequivocal proof? Repeat, Watson, Boveri or Sutton, or Hershey and Chase. So Hershey and Chase will be the correct answer for this question. So we know that Hershey and Chase proved that DNA is a basic genetic material in 1952 by using the isotope P32 and S35 with the T2 type phage bacteria, concluded that DNA is the genetic material. Moving towards the next question, it is saying that the net electric charge on DNA and histone is positive and negative, negative and positive, both A and B, or zero. So basically, if we want to explain this, that DNA consists of a nitrogenous base, pentose sugar, and a phosphate group. DNA has the negative charge due to the presence of PO43, that is the phosphate group. So DNA is negative. Now is histone. Histones are rich in the basic amino acid residue lysine and arginine, which carry positive charges in their side chain. Therefore, histones are positively charged. So DNA is negative and histone is positive. So second option will be a correct answer for this question. Moving toward the next question, 121. In lac operon model, lactose molecules function as inducer which bind with the operator gene, repressor which bind with the operator gene, inducer which bind with the repressor protein, co-repressor which bind with repressor protein. So in lac operon, the lactose molecules functions as inducers and which bind to the repressor protein. So third option will be your correct answer for this question. So why this lactose we are calling inducer? So suppose it is binding with the repressor protein. So suppose this is the repressor protein and this is the lactose molecule. So when it will bind, it will deactivate or it will suppress the repressor, and the enzyme will be activated. That's why it is calling as inducer. So third option will be a correct answer for this question. Moving toward the next question, 122. It is saying that which of the following is correct? Henkin discovered Y chromosome. Drosophila also shows XX, XY sex determination like human. Birds have ZZ, ZW sex determination, while females are ZZ and males are ZW. Grasshoppers show XX, XY sex determination. So we will read the statement one by one and try to explain which is the correct statement. First is saying that Henkin discovered Y chromosome. This is not correct because Henkin discovered the X chromosome while studying on insects in early 1890s. Now the next is Drosophila also shows XX, XY sex determination like human, which is absolutely correct. And in Drosophila, XX is considered as female and XY is considered as male. And we know that in Drosophila we also have X is to A ratio. Here A is the autosome, and this will determine whether this is female or male. Next, birds. So basically what happens is birds, that this is correct, that it has ZW sex determination, but where females here ZZ, that means homogametic, and males are heterogametic. But in bird this is not the case. It is just the exact opposite of us or human. This is not a correct statement. Here ZZ is male, ZZ is male, and ZW heterogametic is female. The last is saying that grasshoppers show XXXY sex determination. So in grasshopper there is no Y chromosome. So a grasshopper with X chromosome symbolized as XO is basically a male, and while grasshopper with 2X chromosome XX is normally a female. This is also a wrong statement. So here second option will be a correct answer for this question. Now moving toward the next question, 123. How many hotspots of biodiversity in the world have been identified till date by Norman Mayer? 17, 25, 34 or 40? So the correct option for this will be 34. It is third option. The biodiversity hotspots basically are a method to identify those regions of world where attention is needed to address biodiversity loss and guide investments in conservation. तो बेसिकली ये एक्सप्लेनेशन यहाँ पे दिया हुआ है, आप पढ़ सकते हैं यहाँ से. Currently 34 biodiversity hotspot have been identified. So yeah. So अभी एग्जाम्स के कुछ दिन बचा है, लाइक 1 वीक बचा है शायद. तो ये अब टेस्ट दे रहा है, तो मैं यहाँ पे कुछ चीजें बोलना चाहूंगा. ये जो आप देख रहे हैं इस टाइप के क्वेश्चन, इंफॉर्मेटिव क्वेश्चन, तो एनसीईआरटी के जितने सारे टेबल्स थे, जितने सारे इंफॉर्मेशन थे, आपको सब पता होना चाहिए. आप एक भी टेबल को छोड़ नहीं सकते, एक भी इंफॉर्मेशन को छोड़ नहीं सकते. ऐसा क्वेश्चन अगर आ गया 17, 25, 34, 43 यहाँ पे दिया हुआ है, अगर आप में इस क्वेश्चन में अगर आपको डेटा पता नहीं होगा तो आप यहाँ पे आंसर मार्क नहीं कर पाएंगे. ये क्वेश्चन +4 कैरी करते हैं, 4 मार्क्स कैरी करते हैं. 4 मार्क्स बहुत होते हैं रैंक इधर से उधर लाने के लिए. तो एडवाइस यही रहेगा कि आप अभी कुछ दिन बचा है, नया कुछ भी मत पढ़िए, कुछ भी नया पढ़ने की जरूरत नहीं है. जितना भी आपने पढ़ा है उस पर कॉन्फिडेंस रखिए और क्या कीजिए, रिवाइज कीजिए जितना बार हो सके. ये लास्ट कुछ दिनों में अपना 100% मैक्सिमम एफर्ट दीजिए, जितना भी हो सके. जितना भी एफर्ट दोगे आप, उतना क्या होगा, उतना आपका रैंक इम्प्रूव होगा. ये लास्ट कुछ दिन बचा है, यहाँ पे कोई कंपटीशन नहीं होगा अभी कि ये नया कुछ पढ़ लेंगे तो ये हो जाएगा, वो कुछ वहां से देख लेंगे, कुछ यहाँ से पढ़ लेंगे. अभी जितना पढ़ा है, अगर आपका मार्क्स अच्छे आ रहे थे तो आप अच्छा करोगे, बस रिविजन अच्छा रखो. ये बोलना था. moving towards the next question, 124. Okay. So it is saying that given below are two statements, one is labeled as assertion, another is labeled as reason. Respiratory pathway is considered as an amphibolic pathway. It involves both anabolism and catabolism. So if we put a because here, that respiratory pathway is considered as an amphibolic pathway because it involves both anabolism and catabolism. It is correctly satisfying. यहाँ पे assertion क्या हुआ है कि इस रेस्पिरेटरी पाथवे को एम्फिबोलिक पाथवे क्यों बोला जाता है, क्योंकि यहाँ पे जो प्रोसेस होता है एनाबोलिज्म कैटाबोलिज्म दोनों ही इन्वॉल्व होते हैं. दोनों ही करेक्ट स्टेटमेंट है, तो दोनों ही एक दूसरे को सेटिस्फाई कर रहा है. रीजन करेक्टली वेरीफाई कर रहा है assertion को. तो यहाँ पे हमारा जो आंसर होगा फोर्थ ऑप्शन, बोथ assertion and reason are correct and reason is the correct explanation of assertion. और एक चीज आप यहाँ पे याद रखिएगा, कैटाबोलिज्म मतलब टूटना होता है, एनाबोलिज्म मतलब जोड़ना होता है. तो ये आप याद रखिएगा तो बहुत क्वेश्चन आगे सॉल्व होता रहेगा. छोटा सा ट्रिक. कैटाबोलिज्म मतलब टूटना, एनाबोलिज्म मतलब जोड़ना. Moving toward the next question, 125. It is saying cytochrome C oxidase complex contains cytochrome A, A3, 1 copper center, cytochrome A and 2 copper center, cytochrome A3 and 1 copper center, cytochrome A, A3 and 2 copper center. ये भी बेसिकली एक इंफॉर्मेटिव टाइप के क्वेश्चन थे एंड वी नो दैट cytochrome here cytochrome C oxidase complex means complex 4 refers to cytochrome C oxidase complex. It contains cytochrome A, A3 along with that 2 copper cell. The fourth option will be our correct answer for this question. Moving towards the next question, 126. Given below are the two statements. The first statement is saying sigmoid curve is the characteristic of a living organism growing in a natural environment. Second, it is saying that in geometric growth following the mitotic cell, one daughter cell continues to divide. So we can see that first statement is saying sigmoid curve is a characteristic of living organism growing in a natural environment. This correct statement. Now the next we know that the sigmoid curve is like this kind of sigmoid curve, and this is the characteristic of living organism growing in a natural environment. Now the second statement is saying that in geometric growth following mitotic cell division, only one daughter cell continues to divide. This statement is incorrect because this doesn't occur in geometric growth. It actually occurs in arithmetic growth. It should be arithmetic growth. So first statement is correct, second statement is incorrect. That's why second option will be our correct answer for this question. Moving toward the next question, 127. It is saying, given below are the two statements, one is labeled as assertion and other is labeled as reason. So first assertion is given, antibiotic are chemical substances which are produced by some microbes and can kill or retard the growth of other microbes. ये बिल्कुल सही है यहाँ पे. This is actually correct statement. Now reason यहाँ पे दिया हुआ है और reason is given. Antibiotic was extensively used to treat American soldiers wounded in World War I. So we have to choose between these two. So यहाँ पे second reason जो दिया हुआ है कि antibiotic was extensively used to the treatment of American soldiers wounded in World War II. ये basically wrong statement है. यहाँ पे antibiotic जो use किया गया था वो basically World War II के बाद ही शुरू किया गया था. World War II में ही शुरू किया गया था. और बेसिकली आप यहाँ पे ऐसे भी देख सकते हैं, अगर ये आपको याद भी नहीं है कि वर्ल्ड वॉर फर्स्ट है वर्ल्ड वॉर सेकंड था, तब भी आप इसको करेक्ट कर सकते थे, क्योंकि यहाँ पे रीजन जो दिया हुआ है यहाँ पे कुछ अलग ही बता रहा है. एंटीबायोटिक का फंक्शन बोल रहा है कि एंटीबायोटिक का यूज कहाँ पे हुआ था, एंटीबायोटिक कब यूज किया गया था, लेकिन यहाँ पे एंटीबायोटिक का प्रॉपर्टीज दिया हुआ है कि एंटीबायोटिक जो होता है एसर्शन में, एंटीबायोटिक का केमिकल सब्सटेंसेस व्हिच आर प्रोड्यूस्ड बाय, जो भी है ये प्रॉपर्टी और फंक्शन जो भी दिया हुआ है. यहाँ पे अगर एंटीबायोटिक रीजन में अगर एंटीबायोटिक के बारे में कुछ फंक्शन या उसका कैसे ये एंटीबायोटिक आया या कुछ मतलब उसके रिलेटेड कुछ होता तो यहाँ पे हम देख सकते थे कि असर्शन रीजन क्या होता है, लेकिन यहाँ पे कुछ अलग ही दिया हुआ है. तो अगर आप फिर भी अगर वर्ल्ड वॉर फर्स्ट या सेकंड ये कुछ याद नहीं रहता फिर भी आप इसको करेक्ट कर सकते थे. सो बेसिकली सेकंड ऑप्शन assertion is correct but reason is not correct will be our correct answer for this question. Moving toward the next question, that is 120. So sporozoites are stored in human stomach, mosquito salivary gland, mosquito blood or human intestine. So sporozoites, basically what happens that after 10 to 18 days a form of the parasite called sporozoite migrates to the mosquito salivary gland. When the Anopheles mosquito takes the blood meal on another human, anticoagulant saliva is injected together with the sporozoite which migrate to the liver, thereby beginning a new cycle. So this is given about the new cycle, but this sporozoite is the form of this parasite that migrates to the mosquito salivary gland. The sporozoites are stored in mosquito salivary gland will be our correct answer for this question. If we also read the malarial cycle diagram, then you can also see that in NCERT. Now next is 129. So it is saying that select the correct option and match the column first to column second. So column first is given ants, birds, fishes and beetles. So ant is column second, it will be 20,000. Then birds it will be 1,300. Fishes it will be 28,000. Beetles it will be almost 3 lakh. So the fourth option will be a correct answer for this question. मैंने आपको पहले भी बोला कि आप ऐसे डेटा बेस्ड क्वेश्चन, ये इकोलॉजी का क्वेश्चन है बेसिकली. इकोलॉजी में ऐसे क्वेश्चन आते हैं, बहुत बार आया हुआ है, बहुत बार ऐसे टाइप क्वेश्चन आए हुए हैं प्रीवियस ईयर्स में. तो आप अगर डेटा को याद नहीं करेंगे तो ये क्वेश्चन आपको तो छूट ही जाएगा. एंड्स कितना था, कितना मतलब माइन्यूट सा क्वेश्चन है, जितना छोटा छोटा इन्फॉर्मेशन आपको याद रखना होता है. बेसिकली यही टेस्ट करता है. एनटी आपका यही चीज टेस्ट करेगा, आपका पेशेंट टेस्ट करेगा, आपने कितना प्रिपरेशन किया टेस्ट करेगा. तो बेसिकली आपको रिविजन करना है, या लास्ट कुछ दिन आपको रिविजन करना है. अगर नेक्स्ट मूविंग टुवर्ड्स द नेक्स्ट क्वेश्चन 130. Match the following columns, column first with column second. Mango, National Park, Wildlife Safari Park, hotspot in the world. So basically mango, almost 1,000 varieties. Then National Park. So National Park are on-site conservation, that means it is basically in situ conservation. Wildlife Safari Park is ex situ conservation, hotspot in the world is 34. So matching all these options, we will get that second option will be your correct answer for this question. Moving towards the next question, that is 131. Ecologists have warned that if present rate of extinction of species continues, nearly dash of species on the earth might be swiped out within the next 100 years. Select the option to fill the blank: 25%, 30%, 70% or 50%. So basically the correct answer for this question will be the fourth option, 50%, because in NCERT it is written that ecologists have warned that if present rate of extinction of species continues, nearly half of the species on earth might be wiped out within the next 100 years. Moving towards the next question 130. So it is saying that pyramid of biomass in sea is generally inverted because biomass of fishes is far less than the phytoplankton. Biomass of fishes exceeds that of phytoplankton. Then, due to buoyancy in sea, the mass of fishes become less. Then, pyramid of biomass in sea is always upright. So, according to all the options, if we compare, first of all, let's understand that what is this pyramid of biomass in sea and why this is inverted. So, we know that the pyramid, if we make for sea, it will be like phytoplankton, zooplankton, then fish. These are small fishes, and then large fishes. So, if we now wanna make the pyramid of biomass, so phytoplankton will have the lowest. So it will be like this. So it will be like this. This is for phytoplankton. Then zooplankton will be a little higher. And the small fishes, the biomass of the small fishes will also be a little higher. And lastly, for large fishes, it will be a little bit higher. That's why the pyramid of biomass, the pyramid of number is upright or could be different kind of, or basically it is upright generally because of phytoplankton more in number, then zooplankton, then small fish, and then large fishes. So pyramid of number is upright, but pyramid of biomass is inverted because the biomass of phytoplankton is lesser than that of fishes. That's why the pyramid of biomass in sea is inverted. So second option, biomass of fishes exceeds that of phytoplankton, will be our correct answer for this question. Moving towards the next question 130. It is saying that sexual deceit is an incident during pollination by a wasp to yucca bee to pollinate calotropis species of bee, beetle to ophiris. So basically the Mediterranean orchid ophiris employs sexual deceit to get pollination done by a species of bee. The third option will be our correct answer for this question. And what is this sexual deceit? So sexual deceit basically kind of एक टाइप का धोखा बोल सकते हो. तो कुछ भी किसको अट्रैक्ट करने के लिए उसको लाइक ये जो इस सब बी को अट्रैक्ट करने के लिए ये जो मेरिटेन ऑर्किड ओफाइरिस होता है ये क्या करता है, उसका जो एक लीव्स होता है वो फीमेल टाइप का दिखा देता है. लाइक ऐसे शो होता है कि फीमेल टाइप का हो रहा है, तो मेल जो बी होता है वो अट्रैक्ट होता है और पॉलिनेशन करके जाता है, लेकिन एक्चुअली में वो होता नहीं है फीमेल. सो बेसिकली दिस इज द सेक्सुअल फॉर्म ऑफ लाइक काइंड ऑफ बोल सकते हो. So yeah, moving towards the next question, 130. In some organisms like sorry, बेसिकली यहाँ पे मैंने लीफ बोला होगा तो ये बेसिकली लीफ में फ्लावर को ऐसे कर देता है, फीमेल टाइप का दिखा देता है, मतलब फीमेल टाइप का ऐसे बन जाता है, नॉट लीव्स, फ्लावर. Now moving towards the next question on that. In some organisms like certain fungi and algae, cells undergoing mitosis repeatedly without subsequently undergoing cytokinesis, what would be result from it? Decrease the chromosome number, decrease the DNA molecules, formation of syncytium, or multicellularity. So, what happens that in some organisms, karyokinesis is not followed by cytokinesis, and that means as a result of which multinucleate condition arises leading to the formation of syncytium. So, third option will be our correct answer for this question. Now, moving towards the next question, match the column first with column second. First is mango. Scientific name of mango is Mangifera indica. Then leopard. The scientific name of leopard is Panthera pardus. Then tiger. The scientific name of tiger is Panthera tigris. Then cat is Felis. So considering all these options, you can see that first option will be your correct answer for this question. Moving towards the next question or the next section, section B of botany, that the DNA of E. coli. So the DNA of E. coli, double-stranded and linear, double-stranded and circular, double-stranded and linear, double-stranded and circular. So E. coli is the gram-negative bacteria. We know that this is rod-shaped, motile or non-motile bacteria. E. coli basically contains a double-stranded DNA as its genetic material, and this DNA is not associated with any histone protein, so it refers to as naked DNA. The DNA is circular with no free. So it somewhat looks like this. So this is like the free DNA, double-stranded circular. So our correct option for this question will be second option. Moving towards the next question, 137. The common mode of sexual reproduction in Chlamydomonas is isogamous, anisogamous, oogamous, or hologamous. So in Chlamydomonas, sexual reproduction takes place through isogamy, anisogamy, or oogamy. But read the question carefully. It is saying that the common mode of sexual reproduction in Chlamydomonas. So isogamy is basically the common mode of sexual reproduction in Chlamydomonas. The first option will be our correct answer for this question. Now we will understand the rest options as well. So basically isogamy, what does it mean? That the fusion of similar gametes is the commonest mode of sexual reproduction, and then it is the answer. And then anisogamy is the fusion of a morphologically similar but physiologically different cells, and then oogamy is the function of different gametes. The hologamous is not relevant here. The first option will be a correct answer for this question. Now moving towards the next question, it is saying that the technical term used for the androecium in a flower of China rose is polyadelphous, monoadelphous, diadelphous, or polyandrous. So basically here 138, the stamens may be united into one bunch or one bundle in China rose. That's why it will be called as monoadelphous. The second option will be our correct answer for this question. Moving towards the next question, that is 139. When a flower can be divided into two equal radial halves in any radial plane passing through the center, it is said to be actinomorphic, zygomorphic, heteromorphic, or cyclic. So, first option will be your correct answer for this question, that in symmetry, the flower may be actinomorphic radial symmetry found in mustard that can be divided into equal halves in any radial plane passing through the center. First option will be your correct answer. Moving towards the next question, that is 140, that Parenchyma cells have thickened corners due to deposition of cellulose, hemicellulose, pectin, or all of these. So, parenchyma have cellulose, hemicellulose as well as pectin. So, fourth option will be our correct answer for this question. That because deposition of this cellulose, hemicellulose, and pectin, they have thickened corners due to they have thickened corners. So what does parenchyma do? Parenchyma basically is a supporting structure, and it supports stem, leaves, basically support stem, then branches, leaves as well. Basically this is the supporting cell, parenchyma. Fourth option will be the correct answer here. Moving towards the next question, 141. So we have to select the mismatch. Gas vacuole, green bacteria, large center vacuole, plant cell, protist, prokaryotes, methanogens, prokaryotes. So gas vacuoles are found in green bacteria, which is correct. So gas vacuoles are aggregate of gas vesicles and are found in green bacteria to impart ions. Then large central vacuole found in plant cell, which is absolutely correct, that the presence of large central vacuole is characteristic of plant cell, while animal cells have multiple small vacuole. Then the next, protist, it is saying prokaryotes, which is not correct. So protists are diverse single cellular eukaryotic organism and belong to third domain eukaryote. And the next is methanogens, which is saying that prokaryotes. It is absolutely correct that methanogens are prokaryotic anaerobes that produce methane gas and belong to domain archaea. So here, third option will be your correct answer for this question. Moving towards the next question, 142. It is saying that which of the following cellular organelle is the site of formation of adenosine triphosphate? Peroxisome, mitochondria, Golgi apparatus, or chloroplast? Come on, this is the most easiest question on this paper, like whatever you are solving. It is saying that which cellular organelle is the site of formation of ATP, adenosine triphosphate. We know that from class 6 class, you know that mitochondria is the powerhouse of this cell. Second option will be the correct answer for this question. Okay, so moving towards the next question, 143. Chromosome decondense into diffuse chromatin at the end of telophase, at the beginning of prophase, at the end of interphase, or at the end of metaphase. Basically, what happens that at the beginning of the final stage of this mitosis, or we can say that the telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. So, first option, that at the end of telophase or beginning of the final stage of mitosis will be your correct answer for this question. First option. Moving towards the next question, 144. It is saying that equational division is usually restricted to haploid cell, diploid cell, prokaryotic cell, or eukaryotic cell. So basically, mitosis of the equational division is usually restricted to diploid cell only. So second option will be our correct answer for this question. What does this equational division means? Equational division basically means that the number of chromosomes will be same. The number of chromosomes will be same from parent to offspring. From parent to offspring. That's why it is called equational division. Moving towards the next question, that is 145. It is saying that apoptotic embryos in citrus arises from synergids, maternal sporophytic tissue in ovule, antipodal cell, or diploid egg. So first, let us understand what does this apomixis or apoptotic embryo means. So basically, apomixis refers to a asexual reproduction in which seeds are formed without fertilization. So in apomixis, there is no fertilization. Apoptotic seeds can be developed in various ways. In citrus, what happens that apoptotic embryos are developed from a maternal sporophytic tissue that is present in the ovule. Each ovule contains many embryo and polyembryony in case of citrus. So here second option is your correct answer for this question. Moving towards the next question, that is an organism with two non-identical alleles is dominant, hybrid, heterozygous or homozygous. So there is no information is given about like which is dominant, which is recessive. So if it is dominant, can't be the answer. Hybrid also can't be the answer, but it is saying that two non-identical, that means heterozygous. The third option will be your correct answer for this question. If the question was given that an organism with two identical alleles, then it should be homozygous. Moving towards the next question, 147. It is saying that lack of independent assortment of two genes A and D in fruit fly Drosophila is due to repulsion, recombination, linkage, or crossing over. The third option will be the correct answer for this question. Let's understand that this like in explanation what is given. And it is saying that Mendel's law of independent assortment states that when the parent differs from each other in two or more pairs of contrasting characters, the inheritance of one pair of factor is independent of this other. For the character to assert independently, they should be located on separated non-homologous chromosomes. But what happens that in linkage, what happens that genes that present on the same chromosome show linkage. It means that these characters remain together and thus low numbers of combination are formed. This phenomena is called linkage, and such genes are called linked genes. So suppose this is a gene and this is a gene. So if the distance between two genes is very close, then there are more chances of linkage. And suppose this is a gene and this is a gene. So the distance is more, that's why there is a possibility of independent assortment, and this will be free from linkage. And one thing you can remember that linkage is inversely proportional to crossing or inversely proportional to distance as well. So as the distance will less, linkage will be more. So third option will be the correct answer here for this question. Moving towards the next question, 148. It is saying that in DNA, percentage of thymine is 20%, then what will be the percentage of adenine? You know that adenine base pairs with thymine, guanine base pairs with cytosine. So adenine is saying that thymine is given as 20%, so adenine should be 20%. And then it will be 30 and it will be 30 as well. So here first option will be the correct answer for this question. Moving towards the next question, 149. We have to select the correct match. Alex Zephyr, Streptococcus pneumoniae, Alfred Hershey and Martha Chase, TMB, Matthew Michelson and F. Stoll, Rhizome sativum, then Francis Jacob and Jacques Monod Lacoper. So this is fourth option is the correct answer for this question. Now we will understand the other options as well. Alex Jeffrey invented the DNA fingerprinting technique. So this is not associated with Streptococcus pneumoniae. Then Alfred Hershey and Martha Chase proved that DNA is a genetic material using T2 bacteriophage. T2 bacteriophage, not EM. Then lastly, Matthew Michelson and Stoll proved semi-conservative mode of replication in Bacterium E. coli. Rhizome sativum was used by Mendel, not Michelson and Stoll. But Jacob and Monod give Lacoper. So Fourth option is your correct answer for this question. Moving towards the next question. Which of the following is considered a hotspot of biodiversity in India? Aravalli Hills, Western Ghat, Indo-Gangetic Plain, or Eastern Ghats? So biodiversity hotspot, that is Western Ghat. Second option is your correct answer for this question. So this concludes the explanation part from my side for botany, and this is the last test you have given, last full test you have given in this Brahmastra test series from MEMONEET. तो कुछ यहाँ पे ऐड करना चाहूंगा मैं, अभी लास्ट टेस्ट अपने दे दिया, स्कोर जितना भी आ रहा होगा तो मॉक स्कोर उतना मैटर नहीं करता है, लेकिन मैटर भी करता है अगर आपका कॉन्सेप्ट सही है। हो सकता है कि मतलब ऐसा आप बोल सकते हैं कि सर मार्क्स नहीं आ रहे, क्या करें। तो अभी ये कुछ दिन जो है यहाँ पे मैं तो बोलूंगा कुछ नया मत पढ़ो, पहले में जो भी बोला रिविजन करो और चीजों को अच्छे से बार बार देखो, जितना भी हो सके जितने बार भी हो सके अपने माइंड को काम रखो, ज्यादा एक्साइटेड मत हो कि एग्जाम होगा, ज्यादा टेंशन में भी मत रहो कि क्या होगा उस दिन अच्छा ही होगा जो भी होगा। कुछ टिप्स मैं यहाँ पे देना चाहूंगा, एटलीस्ट 2 घंटे पहले एग्जाम सेंटर पे चले जाना, तो फिर क्या होगा कि आपको थोड़ा रेस्ट मिल जाएगा, आपका माइंड मतलब आसपास थोड़ा स्टेबल हो जाएगा, क्या करना होगा क्या नहीं होगा इंस्ट्रक्शनों की दे देंगे जो भी एग्जाम में होंगे। और यहाँ पे जब आप क्वेश्चन पेपर को अटेम्प्ट करोगे तो क्या करना है, क्वेश्चन पेपर को अटेम्प्ट जब करोगे तो एक क्वेश्चन अगर नहीं बन रहा है तो उसके ऊपर टाइम वेस्ट मत करना कि एक क्वेश्चन फिजिक्स का नहीं बन पा रहा है तो 5 मिनट उसी में लगा दिए। उस 5 मिनट को अगर यूज करेगा तो केमिस्ट्री के 5 क्वेश्चन बन सकते हैं या बायो के 10 क्वेश्चन बन सकते हैं। तो आप ट्राई करोगे कि बायो फास्ट करने के लिए जो पैटर्न जनरली रहता है आपका जो मर्जी हो कर सकते हैं। मेरा जब मैंने एग्जाम दिया था तो मैंने क्या किया था कि मैं आपके एक्सपीरियंस कुछ आपको शेयर करना चाहूंगा कि मेरे टाइम पे क्या हुआ था। तो मेरे टाइम पे क्या हुआ था कि फिजिक्स का पेपर बहुत हार्ड आ गया था, लाइक मतलब ठीक ठाक पेपर आ गया था। तो क्या हो रहा, लेकिन जब आप एग्जाम दे रहे हो तो आपको पता नहीं चलता है, क्योंकि मतलब तब तो आप अकेला दे रहे होते हो ना, आपको हार्ड लगता है, बाकियों को हार्ड लगा है कि नहीं लगा है तो आपको पता नहीं चलता। इसीलिए आप क्या करते हो कि मतलब 30 क्वेश्चन अगर बना फिजिक्स के तो 30 क्वेश्चन बना तो सही है, तो मतलब 120 मार्क्स तो आ रहे हैं ना। तो आप क्या करते हो कि ओएमआर मतलब दिख रहा है, मतलब ये भरा भरा नहीं लग रहा है तो आप और कुछ नेगेटिव मार्क कर देते, तो वो गलत है। कि एक मार्क्स जो होता है वो आपका रैंक 1000 से वैरी करवा सकता है, और लास्ट जब सिलेक्शन मतलब एक मार्क भी बहुत ज्यादा होता है। तो ये सब चीज नहीं करना है आपको। और जो ट्रेंड में बोल रहा था कि पहले बायो आप करना, बायो आप ट्राई करना कि मैक्सिमम मैक्सिमम 40 से 45 मिनट में फिनिश करने के लिए, 40 से 45 मिनट। और फिर उसके बाद केमिस्ट्री, केमिस्ट्री के लिए लगभग वही उतना ही 40 मतलब 50 मिनट्स या 55 मिनट्स। और फिर इसके लिए आप टाइम देना लाइक 1 आवर 1 आवर 20 मिनट 1 आवर 30 मिनट और जो भी जितना भी आप टाइम। और बाकी अच्छे से ओएमआर भरना, ओएमआर में गलती अब से नहीं होनी चाहिए, ये बहुत ज्यादा स्टूपिडियस मिस्टेक है। जब मैं एग्जाम दे रहा था तो मेरे से भी गलती हो गया था, मेरा 2 क्वेश्चन ऐसे गलत हो गया था तो 10 मार्क्स गए थे मेरे। तो 10 मार्क्स के लिए मतलब जो कॉलेज मुझे अच्छे मिलने थे और वो नहीं मिल पाया। अभी ठीक है सब यहाँ पे स्टेबल है, लेकिन फिर भी एक मतलब वो दुख होता है कि बस क्वेश्चन आपका आंसर पता है, सब कुछ पता है, आपने आंसर निकल लिया, टाइम दे दिया उसके ऊपर, लेकिन ओएमआर में गलती कर दी। ये मतलब जब घर आओगे और आंसर मिलाओगे ना तो आपको ये बात जचेगा नहीं कि यार ये मैंने क्या कर दिया। तो यही चीज, कुछ सिली मिस्टेक मत करना यार, एक भी सिली मिस्टेक नहीं होना चाहिए। कि जो क्वेश्चन हार्ड है वो आप अगर आपका प्रिपरेशन अच्छी है तो वो सबके लिए हार्ड है, सिर्फ आपके लिए हार्ड नहीं है, वो सबके लिए हार्ड है। तो आप एक काम करना कि जितना भी बन पाएगा, जितना भी बन पाएगा, जो सही बन पाएगा वही बनाना। उसमें भी गलती होंगे लेकिन वो मतलब एक्सेप्टेबल है। लेकिन जो चीज आपको एकदम नहीं आ रहा है, एकदम मतलब आपको कुछ अता पता नहीं है, अपने पढ़ाई नहीं है, तो लाइक वो उस क्वेश्चन को अटेम्प्ट करने का कोई मतलब नहीं बनता, और 90% टाइम नेगेटिव मार्क्स से देके जाएगा। तो यही चीज आपको कुछ कुछ चीज आपको ध्यान रखना है। और आई होप अगर आपने अच्छी प्रिपरेशन किया हो, सब कुछ ठीक ठाक किया हो, तो एग्जाम बहुत अच्छा होगा, बहुत अच्छा होगा। ऑल द बेस्ट फ्रॉम माय साइड।